Now we are going to start today's second session of plenary lectures and discussion lectures. We have three distinguished speakers in this session, and I'm honored to introduce our first speaker, Professor Paul. He's professor at the Department of Biosciences, UK. Professor Paul is working in the area of molecular biology, cell biology, parasitology, and lipid biochemistry. Professor Paul undertook his PhD studies at the MRC National Institute for Medical Research and focused his research work on the residual classes of epicomplex and protozoa. While doing many projects at the Imperial College, he developed interest in the molecular, biochemical, cellular biology of kinetoplastic protozoan parasites and lipid biosynthesis area. Professor Paul is the founding director of Durham University Center for Global Infectious Disease and director and principal investigator of the MRC DPRF Global Network for NDC. Professor Paul has multidisciplinary approaches to study and exploit novel drug targets within both kinetoplasmids and epicomplex and parasites. Now I welcome Professor Paul, and the title of the presentation is Natural Product Antileishmanials.
Thanks for that kind introduction. The screen come up. Okay, thank you, and thank you for the invitation to speak today into this fantastic audience, both here and remotely online. So, yeah, pleasures and pitfalls. It's not an easy road in any way, looking for new potential drugs, either natural products or small molecules. And I'll tell you a bit about that in the next 35 minutes or so. So, as said, I'm in the Department of Biosciences in the group Biomolecular Interactions, which MCO also occupies, where we work closely together. And also, as was mentioned, I do lead the Global Network for Neglected Tropical Diseases, of which this institute is a very, very important partner. And Iqbal Samra and I will be discussing some future work within this network. It's funded through the Global Challenges Research Fund, which is a development aid program, which runs until this summer, essentially. Okay, sphingolipids are important lipid species in all eukaryotic organisms. Probably best known for forming these microdomains known as lipid rafts, characterized here by Kai Siemens many years ago, mid-90s. Implicated in almost everything in eukaryotic cells. So my interest is particularly sphingolipids in protozoan parasites. This includes the parasite toxoplasma, which you can regard as a model apicomplexer. It's related to the Plasmodium species, which causes malaria, of course. Definitive host is the cat, which you can see there. But importantly, it causes neural damage in particularly in humans, but also ocular damage. Also, and this will be interest around the world, including in Pakistan, a great cost to the sheep farming industry with the abortion of lambs. Anyway, I'm not really here to talk about toxoplasma in particular, just to mention that we have worked on sphingolipid biosynthesis in toxoplasma. It's very unusual in many ways. This is the first enzyme, serine palmitoyl transferase, SPT, in eukaryotes, and in all eukaryotes, aside from the apicomplexer, it has a structure of two subunits, a catalytic one, LCB2, and a non-catalytic regulatory one, LCB2. In contrast, the bacterial enzyme, which is not an ortholog, it's a functional equivalent, very different evolutionary origin. However, the toxoplasma, Plasmodium, and other apicomplexer harbor an enzyme which is very similar, and then we've published this, and you see Emka and I have worked closely on this project. Okay, but leishmaniasis, important problem in Pakistan and elsewhere. Iqbal was telling me there's a large outbreak in Sindh just in the past few weeks. It's on the increase across the world, endemic in over 80 countries across 4 continents, well over 1 million new cases, probably massive underreporting, well over 12 million people infected at any one time, anywhere between 350 million and 1 billion people at risk, and a priority disease. So this is something I picked up in the newspaper on a visit to India about 3 years ago or so. Polarised, black fever, visceral leishmaniasis is still a problem in Bihar, particularly in northeast India. But this I want to really mention because this relates to the outbreak in Sindh as well. This is something I was lucky enough in 2019 coming here to visit the Jinnah Hospital and the dermatology department and actually meet these children and their father come back from being in a rural area, and the children had all got these manifestations of sores, some of which were responding to treatment, some of which were not. So for those of you who don't know, leishmaniasis is caused by protozoan parasites of Leishmania species, sandfly vector, these flagellated promastigote forms, bite a human or another mammal even. There's also animal reservoirs involved. They transform to amastigote forms and cause this manifestation of cutaneous, mucocutaneous or visceral disease, cholera. Okay, and then the cycle continues. So we're dealing with a complex vector-borne disease with animal reservoirs, very difficult to control and deal with. So current treatments, this is a very, very limited arsenal of treatments. Antileishmanials here, they're the mainstay of treatment for cutaneous leishmaniasis. This is what you would be given if you went to the Jinnah Hospital and received treatment, an injection of these directly into the lesion or systemically. Liposomal ampicillin B is difficult to access, really is used for visceral leishmaniasis primarily, but in that it's supported by WHO. And paramamycin, rather less used, a second line therapy. But you see both of these are natural products themselves. You can see that characteristic natural product complexity. Pentamidine is less used and it's quite toxic, so it's really on the back burner at this point. Now, all of these are injectables, not already available. It's only miltefosine, which is used for visceral disease, which is actually already available. So, terrible selection of drugs. So, our interest at Durham is really understanding the mode of action of drugs and trying to inform the process of development. So, including the miltefosine I was just mentioning. And what we know about miltefosine, it does have great complex effects on sphingolipid and sterol metabolism in the parasite as well. So sphingolipids. So the core sphingolipid is ceramide, which you can see at the top there. Sphingomyelin is what we have, and other mammals and other animals indeed. So you can see the phosphocholine head group. However, Leishmania, yeast and plants all have the phosphoinositol group, which you can see here, which confers rather different properties biophysically on this complex sphingolipid. So what you can see here is that serine palmitoyltransferase activity, if not structure, is conserved across the eukaryota, but then we get this dichotomy between a sphingomyelin synthase making sphingomyelin in humans, for example, and yeast plants and at least some protozoa using inositol phosphoryl ceramide synthase, IPC synthase, to make IPC. So quite different, a dichotomy. So the theory of targeting this is that we can get specific inhibition that doesn't harm the human equivalent enzyme. So the enzyme sits at a cusp of life and death, controlling the quantity of diacylglycerol, which is a mitogenic, growth promoting agent, and also the quantity of ceramide, which is apoptotic agent. So it really sits in the balance between here. So if we interfere with the function of this, we will interfere with the balance, the seesaw of life and death in the cell. It is a druggable target. Fungi have been studied extensively, and there's again several natural product antifungals have been identified that specifically target this enzyme. I'll highlight this one here, peptide or ibacidin A, because I'll come back to that. Complex molecule. It is subject to some development in industry, hasn't reached the clinic for fungal infection yet. So, high throughput screening for drug discovery. You've probably Igal mentioned several elements to this, and Emka then followed that up. So we have conventional target-based screening with a target that you've identified and validated, a typical biochemical assay. Some advantages, it can be simple. You can really understand the way the agent, as Emka showed, interacts with the protein. But the limitations, you need the protein, you need the validation, you need the assay. That's not easy necessarily. Iqbal mentioned phenotypic high throughput screening. This has some advantages because you don't need that target. You're looking in a cellular context and you're screening everything at once. Disadvantages can be insensitive for looking for a small molecule that you can develop. It can be very expensive as well because of the culture problems that you can have with certain pathogens or cell types. And something that you may be less familiar with is a combination of the two that we've worked on, that's yeast-based high throughput screening, where you take the target and you construct an assay in the yeast cell. Again, you don't need a purified target, and you then screen it in a eukaryotic cellular context. Yeast are cheap, so it has numbers of advantages, and you can develop fairly sophisticated assays. Disadvantages is that yeast are very robust and tough, actually, with their cell walls, so they can be very sensitive. And you have to rely on the protein being actually functional in what is a foreign system. Yeast, of course, is not the pathogen we want to screen for. So just generically, I'll be very quick in going through this. So we need a valid assay. This doesn't just apply to yeast-based assays. This is for all assays, whether you're screening against cell types in a phenotypic sense or a target-directed screen. So we need to consider the validity. So we need a valid target, preferably one that's been validated genetically by knockout studies. And we need to, if we're looking at a phenotypic assay, we really want to try and look at the appropriate cell type. Leishmania, that's that intracellular amastigote form, which causes the disease in the human or animal. We need to consider cost as well. There's no point having an assay that is just going to be basically too expensive to use. A very important thing, and this relates here to a cellular assay, we need to think about linearity. So this is our cell readout, and this is the number of cells. So this is a proxy for a typical assay. We want it to look like this. So the readout is equivalent to the number of cells, not this curve where it breaks down and we cannot really tell what's going on. And a big thing at the moment, a big thing for me personally, is reproducibility and transportability. There's no point me having an assay if I can't take it from Durham to Karachi and it works there as well. So how do we validate the assay? We use something called Z prime or Z factor, and essentially we look at the standard deviation and the difference between a positive control, which is a known inhibitor or drug, and a negative control, which is our untreated assay. So the bigger the difference between the positive and negative, and the smaller the standard deviation, the nearer Z prime, Z factor is to 1. This is a piece of real data, positive and negative control, standard deviation, mean. We just put that in and we get a Z factor of 0.92. So that is a very robust assay. In industry, anything over 0.5 is regarded as robust. Okay, so taking those factors into consideration, I'm just going to talk about work we've done, which has been long published now, on the Leishmania IPC synthase to try and look for specific inhibitors. Now, this is a transmembrane enzyme, so EMCA would not want to try and crystallize this. It's very, very difficult to actually formulate an assay. We have a biochemical assay. Actually, we have two now, but they're very expensive and very difficult. So we decided to use the yeast-based system because, as I mentioned, IPC synthase functionality exists in yeast and it exists in Leishmania. So we can quite happily engineer the yeast to be reliant on the Leishmania enzyme, as you can see here. So this is the yeast expressing the Leishmania IPC synthase instead of its own, and it's entirely reliant on the expression of that proteasome enzyme. So what do we do at this point? So we go through a whole series of validation steps. So this was actually trained with a small library of 10,000 compounds. We looked at the Z prime to make sure the assay was robust and reproducible across plates. And then we went from 96 to 384 well plates to 1536 well plates. So these nanoliter level plates, because ultimately, working with GSK Trescantos in Madrid, we wanted to screen the entirety of their library, which is about 2 million compounds. So it's not as big as a virtual library, but it is big if you're going to consider actually a screening assay where you screen in a relatively short space of time 2 million compounds. So we have the assay, it was robust, it was miniaturized. Remember, we need a control as well. So our control was the same assay, but with the yeast enzyme. So essentially, we're looking at differential activity against the Leishmania enzyme compared with the yeast enzyme we've replaced it with. So 2 million compounds to start. So as a primary screen, we look at a single concentration just against the Leishmania enzyme in the yeast system. So it's a very pragmatic process when you're working with industry. So we go from 2 million, and we just change the parameters, or at least GSK changed the parameters to identify 20,000 compounds. And that's kind of the amount that they can deal with in the next stage, which is looking at two concentrations and the Leishmania enzyme compared with the control. All one is the yeast IPC synthase. So we're looking for that differential effect. So at this point, we went from 20,000 to 4,208 compounds. And it was those, and again, it's pragmatic. The parameters were shifted around until we got about 4,000 compounds. Then we go into a much more intensive screening process, which is dose response in duplicate against the Leishmania enzyme and the yeast all-one IPC synthase as well. So at this point, what we're really looking for is very specific and very active compounds against the Leishmania enzyme. That's our readout. So obviously we don't want non-active compounds. We don't want those that are active against the yeast enzyme, but not active against the Leishmania enzyme. I mean, these are interesting, don't get me wrong, but they're really not what we wanted to look for. Now, particularly, this is very, very important in this kind of yeast-based system, a cellular assay. We don't want compounds that are active against both enzymes, because all that's telling us probably is that we've got some antifungals, which we know we've got antifungals in that library. That's a fact. So we're really looking for these. We're looking for those that are active against the Leishmania enzyme, but not the yeast system. So we're very, very focused on these, and we're looking for those that are most active and most specific. So this is the combination of those 4,208 compounds in dose response. Log scale, so 6 is log 10.6, so it's a micromolar. So what you can see here is the Leishmania assay along the bottom here and against the yeast enzyme here. So as we go towards the bottom right-hand corner, we are getting those that have high efficacy against the Leishmania system, but also high selectivity. So they're the ones that are in orange, misty blue, and brown, you can see, just in this corner. So again, in log scale, we're looking at the ratio. And just thinking about that corner there, there's 500 compounds that have a ratio of about 30-fold selectivity in log value for the Leishmania enzyme. So we've got highly selective and highly potent compounds against the enzyme in our system. Okay, so we've gone through this whole process from 2 million down to 500 hits, exactly 500, active and specific. And then we go further, again, using an industrial algorithm, really to cluster the compounds so we don't have a lot of repetition in our subsequent analysis. We don't look at 30 compounds which are highly related. So we just take pairs of compounds which are similar, and of course we keep the singletons as well. And then we go from 500 to 211. So that's an amount that at least I can get my head around analysis process as well. So let me actually, it's the first time that we go and hit the cells at this point. So we look at azenicae mastigotes. So these are Leishmania mastigotes which are grown in the absence of the host cell, azenically. And the hit criteria, you can see here, an activity, again a log value, effective concentration 50, knocks down the growth by 50%, greater or equal to 5, which means there have to be an EC50 of at least 10 micromolar for us to take forward. The product formulation index is an algorithm which indicates developability, so that's based on size, number of aromatic rings, which we also look at separately, various phys-chem properties that indicate the ability to develop any hits further towards drugs, essentially. And also, of course, we look at selectivity against mammalian cells. Classically here, the liver cells, HepG2. And we want them to be at least 10-fold more active against the parasite than the HepG2 cells. So at this point, we go from 200-ish to 25 hits. Now, this when it comes back to what I was saying, is that you have to think about the life cycle stage that's most relevant and the one you can actually screen against. None of this is easy, of course, but we look at screening in the macrophage because the Leishmania mastigote exists and multiplies within macrophages in humans and other animals. This is the assay basically we use, actually created and shared by Manu Duraika, who's at Dundee. This is a picture from his paper. It's the same assay we used. This is Leishmania donovani mastigotes within macrophages, and using fluorescent staining and some smart camera work, they can get a relatively rapid idea of the number of mastigotes that are infected, such as this one, and also the number of mastigotes within each macrophage. So similar criteria, we're looking for at least 10 micromolar activity, and the selectivity at this point, we want to have something that's selective compared with against the macrophage, THP1 being a cancerous human macrophage line that we use in such experiments. So 25 compounds, and we end up at this point with 5 hits, which include 2 series, which means 2 pairs, and 1 singleton. Now this is an audience of chemists. You'll know this is a benzazepine. You can see down here, and this is what we took forward for further development. Now, very, very important. I mentioned how difficult the enzyme assay, the biochemical assay is, and it's difficult and expensive in time and money. But we wanted to validate our compounds against the enzyme in vitro. We do actually do that here, and we formulated the assay in a 96-well plate, and we validated that the identified benzazepanes and some derivatives are both submicromolar inhibitors of the enzyme in our assay. Very important. Now, the pitfall, this is not natural product discovery, but it applies to natural products, is that we then were able to take this into what was the Wellcome Trust drug discovery engine, again with GSK at Trascantos. Harsh milestone. We need a lead within 4 months. That's at least 70% effective. That's before you start development. This is 70% effective in an animal model. We failed. So we should never be ashamed or embarrassed of failure. We just have to learn from it. And drug discovery is a path of failure to a large extent. So we failed, but we published. We've also looked, and I'm glad Iqbal mentioned these things, and I'll come back to some similarities later on. Tamoxifen also inhibits the enzyme, which we've looked at with Silvia Uliana in São Paulo in Brazil. And also with my colleague Patrick Steele and Bartira Rossi Bergman. Patrick's at Durham, Bartira's in Rio de Janeiro. We've also identified clemastine, fumarate, and antihistamine as an inhibitor of the enzyme. And actually, this has gone through into preclinical trials and is currently being developed by Patrick and others in a fairly large-scale program. Okay, so HGS for drug discovery. I've spoken about yeast-based HGS, which is essentially a variant of a target-based approach. I'll come back to the phenotypic process. So this was work published by colleagues at GSK, where they looked at their entire library, again, in phenotypic assays against Leishmania donovani and 2 Trypanosoma species. And they created boxes of hits. So this was using a phenotypic approach, which has become increasingly popular. Now, if I'm going to now spend the next 15 minutes finishing off and thinking about natural products as anti-parasitic, great examples. This is the Nobel Prize in Physiology and Medicine. Campbell and Amura for ivermectin, becoming ivermectin, the nematode therapy used in humans and animals, and UU2 for the identification of artemisinin, which has transformed the treatment for malaria. So both natural products, both highly important, widely used drugs. Both rather complicated, as you can see from this. So back to aureobacidin, just very, very briefly. As I mentioned, aureobacidin is an antifungal. It's a fungal-produced antifungal. And this company here in Michigan okay, well, hopefully it won't cut out as much now. Aureogen have worked on semi-synthetic processes to produce libraries of aureobacidin, and we've worked with them with toxoplasma, which I mentioned before. And the thing about toxoplasma, there's no treatment for this encysted form, which exists in brain and muscle tissue, which is responsible for many, many problems, and perhaps even mental health disorders. So we've focused on that, and we've looked at aureobacidin and some of these analogues, one of which you can see here, has a similar effect, and they're both active against the encysted form, at least in vitro. We haven't looked at this further in animal models. Okay. So back to leishmaniasis. I already mentioned that amphotericin B and paramomycin are natural products, which is pretty obvious when we look at them on this list of compounds. So again, some industrial work with a company called Hyperglycaemia in the southeast of England. So they have a fungal extract library. So these are not pure compounds, but they're secondary metabolites, so drug-like in many respects, low molecular weight, for example, so ripe for development. So we took a screening approach of these 313 fungal extracts from all over the world. It could be from the back garden in London to the Amazon rainforest, and screening them against azelic macrophages of Leishmania mexicana, and also mammalian cells for cytotoxicity. I'll just use this. This won't move around as much. So using these, we've got 38 potent hits at, these are mixtures, remember, 23 selective, more than 48-fold. And then after a typical industrial natural product process of subtraction, IP issues and dereplication to avoid following down rabbit holes, we go through primary fractionation using HPLC. So it's a lot of data summarized in a very complex bar graph, but essentially we see some holes in this, where these holes are is where the fractions are active. So one of these, stratal B, was previously reported as an antileishmanial, so that gives us proof of concept. So we're following the right rabbit hole in this case. And then we go to secondary screening, because we're still in mixtures in this stage. So this is the secondary screening process. We did take some of these forward to tertiary screening, but all activity was lost. Not an uncommon thing, I am told, in natural products. But if we look here in panel G, what we can see is a perfect inhibition of growth of the amastigoleishmania. And this is this compound in the secondary fraction. It is pure HD 8711. Okay, so this is what we took forward. So as I mentioned in the last example I gave, we have the azenic screen, but we do need to look at the activity in an infected cell model. So the azenic line is the red line. The INMAC, the infected cell model, is the black line, which you can see here. So you see the EC50 is almost identical. And then, of course, the toxicity, the uninfected macrophage, is the blue line. So we have a selectivity index of 5.5, which is marginal, to be honest, but so we haven't taken this further. Maybe this is another pitfall in the natural products and the drug discovery world per se. What we see here is we have this high degree of oxygenation and unsaturation within this after we elucidated the structure with mass spec and NMR. So what we think is a highly reactive molecule. Now, this is where we can look for target deconvolution with our colleagues led by Mike Barrett at Glasgow University. So we use a polyomic approach. So we're looking at changes to the metabolome in this case. So this uses a 36-hour time point, dosing the cells at 2 times the EC50. And essentially we get a number of clear changes, some of which I've highlighted in bold. So we've got cysteine appearing, and also a decrease in trypanothione and various forms of trypanothione you can see here. Now, let's explain what this means or might mean. Trypanothione is central, which you can see on the bottom here, in the trypanothione peroxidase system, into the ability of the parasite to deal with oxidative stress. Okay, it's the redox system in the parasite. So the fact that we're having this oxidized cysteine derivative, cysteine, in the treated cells, and we're getting decreases in trypanothione, and indeed the precursor glutathione, which is something we see in other systems as well, interferes with this system, and we get oxidative stress. Now, it's not the best data because the peaks are poorly resolved. That's the limitation of using a broad-based metabolomic approach. However, when we look at the molecule a little bit further, we can see these unsaturated ketone moieties, and really this suggests that they're Michael acceptors, and they're very likely to have a very active and reactive nature towards nucleophiles such as trypanothione and glutathione. So yes, they're active, but you can see also why it's toxic. So again, we published this, and I just want to briefly mention work we've done on semi-synthetic saponins with a small company in Wales, Natural. In some ways, these are more exciting because they're very benign. Iqbal mentioned the steroidal derivatives. These are steroidal compounds as well, terpenoids, and with just this disuccinate addition to this core, we get selectivity against the parasite compared with the host cell. Otherwise, saponins, as some of you will know, are just generically toxic. So we're following this up currently, looking at formulations because it was non-active in our intracellular macrophage assay. And another example, getting back to the trypanothione system, is chalcones, which is another one of these highly reactive, unsaturated class of natural products. Again, work with Patrick and also including Emka here. Patrick used a chemical approach to make this into a probe, and then together we identified that it bound to the trypanoredoxin peroxidase, a key enzyme in that trypanothione system I showed you a few slides ago. So, that's another approach to looking at targets, is to derivatize. This is a chemistry favorite approach. Derivatize the compound and look for binding to the target. I would say this also binds to lots of other targets as well. So you have to be aware of the problems that could lead to. Okay, so I'm just going to finish off in the next 2 minutes just by talking about target validation. So this is what our GCRF network is all about, really, because we're trying to fill a hole that we have in the development of Antileishmanials, really by getting solid identification and validation of drug targets. So downstream, we can start to think about drug discovery. So on a personal level, and this does involve Iqbal as well as many other collaborators from across the network, we have actually secured further funding from Tres Cantos Open Lab jointly with the network to actually look at some of the compounds that came out of that phenotypic screen that I showed you earlier that GSK led to deconvolute the mode of action using some of the approaches I mentioned, plus some genomic approaches as well. Okay, so I just want to finish off. I mentioned our focus on cutaneous leishmaniasis. So this is a video that we made
Did you
recently.
know that poverty and risks from many neglected tropical diseases are interlinked? Many of these diseases can be difficult to recognise, yet they often cause serious societal and community problems. Let's look at the example of cutaneous leishmaniasis. Selina and her family live in Karachi. They have just returned home from visiting their grandparents in rural Pakistan, where deforestation and poor sanitation have created ideal conditions for sandflies. These tiny biting insects carry several types of leishmania parasites. Selina and her brothers became infected whilst they were playing outside. Skin-based leishmaniasis causes painful, crusted, open sores. The children's father decides to take them to General Hospital. Treatment is painful, and around a third of the infections don't respond to the drug at all. Because the children's father loses a day's wage each time he takes his children into hospital for their treatment, the family has to cut back on food in order to meet the rent, and often go hungry. This is not an uncommon story. There are more than 1 million new patients with cutaneous leishmaniasis each year, and around 40 million survivors carrying scars, causing serious physical and mental health problems. Ignorance surrounding this neglected tropical disease often causes sufferers to be excluded from education or employment opportunities. In some cultures, women and girls with these scars are considered unmarriageable or even forcibly separated from their children. After many painful weeks of treatment, Selima's brother's sores have healed but have left unsightly scars. Selima is not as fortunate, and her terrible sores remain. She is reluctant to go outside and believes her life is over. Diseases of poverty can be overcome, but there is much work to be done. We are working towards better understanding, more patient-centered care, and more consideration for the families affected. Yet, there is an urgent need for more research into better diagnostics, treatments, and vaccines to bring new hope for people like Selima. To find out more, visit the links on screen.
Okay, thank you. Of course, that's a story that, yeah.
Nice presentation, and thank you for sharing your excitement. Now the session is open for question and answers. We have 5 minutes now, 4 minutes, so we can take 2 questions now.
Thank you very much. Thank you very much for your lecture. I was wondering if you about the Sphingolipids role in the fashion. I mean, what was the role of the metabolite? I couldn't catch your point.
Okay, about the oxidation. Yeah. I'll just briefly go back to just so I get the right slide. Here. Is this what you mean?
Sphingolipids, like ceramic.
Oh, okay. Yeah, all right. Well, yeah, the sphingolipids. So the sphingolipids are structural components of the plasma membrane, but they're also involved in forming and acting as signal transduction complexes and secondary messengers. So that's the first thing. So interrupting sphingolipid biosynthesis is lethal in most systems, not all, most. And the second point is because of where the enzyme sits, it's substrate. So the use of ceramide, ceramide gets removed to form these complex sphingolipids. And ceramide's pro-apoptotic. So if we inhibit the enzyme, we get an increase in ceramide because the enzyme is not using it. We've measured that, and that's what happens. And we also get a decrease in diacylglycerol. That's a bit more complicated. We don't really know in leishmania what that does, but in eukaryotic, higher eukaryotic cells, it's mitogenic, so it enhances cell growth. So we're kind of increasing ceramide by inhibition and decreasing diacylglycerol. So we are increasing cell death and decreasing cell growth.
I have seen some papers that discuss enzymatic activity against ceramide by testing and stuff that is the cause of that we are testing against some causing some activity against cancer lines. So I thought that maybe certain
yeah, I mean, I think ceramide, the role of ceramide is fairly well understood in mammals. Fairly well understood, not very well understood. It's not well understood at all in protozoa at this point.
Excuse me, sir. I have a question. Sir, I have a question. Sir, why is vaccine not effective against such tropical diseases? Vaccine technology.
Yeah, well, I think we're discussing this over breakfast actually this morning, and I think we're due to see some changes. I suppose the most significant example is the malaria vaccine, although the efficacy of that is very low to prevent disease, but it does prevent death in small children. Leishmania might be more complex as a disease of the immune system, although the learnings of the last 2 years on the rapid development of vaccines from new technology using RNA, for example, shows that we can do this very quickly. But also, the most importantly, it shows it does take hundreds of billions of pounds and a lot of global government effort. So if the global government effort was there, then maybe, just maybe, Leishmania is possible to have a vaccine and deal with the problem. But another point is it's an epidemic disease largely. I mean, it arises and then falls back. So it might be difficult to persuade people they need a vaccine in some circumstances.
Thank you, Professor Paul. So, on behalf of the organizing committee, I give
Thanks for your help.
our next speaker is Dr. Nisar Ahmed. Dr. Nisar is speaker in clinical biochemistry at University of Dr. Nisar Ahmed has received the British Museum Award 2014 for science and engineering, and he is top-cited author for the General Diabetes Research and Clinical Practice in 2013. Dr. Ahmed has also received MMU Teaching Award for Best Supervisor by MMU Students Union. Dr. Ahmed has received his doctoral training at the New York Medical College, USA and University of Birmingham, UK. He is fellow of the Institute of Biomedical Science and board membership of Biochemical Society, Association for Clinical Biochemistry, Diabetes UK, Diabetes and Obesity Research Network. He is the author of over 100 publications, and this includes 6 best-selling textbooks used worldwide on biomedical science programs. These include Biology of Disease, published in 2007, Clinical Biochemistry and Biomedical Research, Practice, both published in 2016 by Oxford University Press and Johnson. His research interests include glycation in diabetes, free radicals, and antioxidants and nutraceuticals. Now, I will invite him for his presentation. The title of his presentation is Pomodical Tensia Inhibits Advanced Glycation and Correct and Promotes Angiogenesis in Vitro, Possible Role in Management of Diabetic Congenital.
Okay, good morning, everybody, and Assalamu alaikum. First of all, I'd like to thank the organizing committee for inviting me to this conference. It's always a delight to come back to Karachi, and usually these are the sort of best days, you know, for me to spend some time here. I'm sure all of you know I've come all the way from, this doesn't seem to be working. Okay, I'll try this. It's not working here, right? Okay, so I've come all the way from Manchester. So this is a city in the northwest of England. It's quite a large city. It's famous for football, and it's where the Industrial Revolution started. and I actually work at the Manchester Metropolitan University. This is a very large modern university. They have about 36,000 students. They're a very multicultural university with people from all over the world. So if any of you youngsters are thinking of going to the UK and you want to have a really good time, then, come to Manchester. If you want to work hard and do really well and so on, then, you could consider Durham, okay? So those are your choices. This is our Faculty of Science and Engineering. I'm telling a lie. It doesn't actually exist at the moment. It's being built, but we will be moving into this early next year, and it's a huge investment by our university. We're spending about £150 million to try and improve our teaching and research facilities. So today's talk, I'm going to talk about a plant called Momordica carrantia. It's also known as bitter gourd. It's also known as bitter melon in some places, and of course in Pakistan we call it Karela. Okay, and I'm going to talk about Momordica carrantia. I'll refer to it as Momordica carrantia. Okay, this uses proper scientific name. So I'm going to talk to you about Momordica carrantia, its use in diabetes, its possible beneficial effects, and I'll share some preliminary data. This is largely in vitro data, which is, we've got results for in my laboratory. And as I said, this fruit is referred here to as an ugly fruit. I don't know why they call it an ugly fruit. I think it's nice. I think it tastes very nice as well, especially when my mum cooks it. Not the case when my wife cooks it, but that's a different story. Okay, so let's kick off by defining what diabetes is. And many of you will have come across this definition. The key word here is hyperglycemia. So it's a disease where the hallmark is a high blood sugar level or hyperglycemia. And nearly all the problems that people with diabetes have is because of this hyperglycemia, as you will see from my lecture. So the whole focus as far as diabetes management is concerned is to try and reduce this hyperglycemia, try and get it back to normal levels, which is not actually easy to do. Okay, as you know, we're in the sort of, we've had a pandemic for the last 2 years, and diabetes was in the media. People who had diabetes and were infected with COVID-19 had a very, very poor outcome. And this hit the headlines. A very high proportion of people who had diabetes sadly lost their lives if they were infected by COVID-19. And what was realized very, very quickly was that the outcomes were much better if your blood sugar was well controlled. But if it was not well controlled, then the outcomes were not so good at all. So again, it goes back to this message of good control of blood sugar levels. And diabetes is a global problem. It's a huge problem, huge health issue worldwide. The International Diabetes Federation has estimated that there were 537 million people affected by diabetes in 2021. And this figure is going to increase to nearly 800 million by the year 2045. So these are huge numbers. If you look at the predicted increase in numbers of diabetics by the year 2030 and look at different countries, then these are the top 10 countries. India is number 1, Pakistan is number 5 on this list, okay? But look at all the countries in red. Now, these are all countries where they often use alternative approaches to manage diabetes, including the use of Momordica carrantia. Now, this is a real tragedy behind diabetes. There were 6.7 million deaths in 2021 because of diabetes. And this is what's been reported by the IDF, right? We are all making a lot of fuss about nearly 6 million deaths because of COVID-19. But this is the number of deaths due to diabetes in 2021. And these deaths, nearly all of them, okay, well, are due to long-term complications. It's the complications that these people develop. That's what actually kills them in most cases. And what we know is that there's an increased risk of getting these chronic complications if you don't control your blood sugar levels. And these complications are the major cause of morbidity and mortality in people with diabetes. What's going to kill you or disable you, essentially. And what happens is you get damage taking place to blood vessels. And this can be large blood vessels, macrovascular lesions, or it can be damage taking place to the fine, smaller blood vessels, in other words, microvascular damage. And the damage happens to various parts of the body. So you could end up having a stroke in the brain, and you could lose your eyesight because of damage to the back of your eyes or to the eye lens. You've got an increased chance of having heart disease, and that's what usually kills people with diabetes. Renal disease, peripheral vascular disease, damage to the nerves, neuropathy, and of course, foot problems. Foot problems are interesting because they're both a combination of macro and microvascular disease. And one of the key things there is impaired wound healing. So if someone's got diabetes, they undergo surgery or they get some sort of wound, it takes a long time for that wound to heal. And that opens you up to the possibility of infections. And if your wounds get infected, and it's usually lower limbs and feet, if they get infected, you can end up with this situation where you have gangrene. Okay, and once you get gangrene, there's not much you can do but amputate the affected part. So it's a real tragedy. Many people with diabetes end up with amputations. They lose their feet or they lose their limbs. I want to focus a bit on wound healing because I'm going to come back to that, because a lot of the studies we did in this project were related to wound healing. Now, wound healing, as I said, is delayed in those people with diabetes. So it's a long-term complication of diabetes. It's quite a complex process, but just to simplify it, there's a number of events that happen. First of all, as soon as you get a wound in your skin, you form a blood clot, and that's necessary. You want to, block that opening because microorganisms will get in and cause an infection. You then get attraction of phagocytic cells to the wound site, and they clear up all the mess. They, take care of any microbes, any dead cells, etc. And this is the key step. You get formation of new blood vessels, and that's known as angiogenesis. It's a crucial step in wound healing. And we know that in diabetic wound healing, angiogenesis is impaired. And I'm going to look at angiogenesis in more detail later on. And finally, you get closure of the wound. And that's what you really want if you want to avoid infections. Let's just focus on angiogenesis. Again, angiogenesis, it involves endothelial cells, and they undergo a number of processes. And there are three key things that happen to these endothelial cells during angiogenesis. First of all, they undergo proliferation, they then undergo migration, and they're also involved in tube formation. And that's what angiogenesis is. And really, these are the sort of three things that we've focused on when we're looking at angiogenesis in our laboratory. Now, the high blood sugar, as I've just mentioned, is very important because if you don't control your blood sugar, you're more likely to get delayed wound healing, impaired wound healing if you happen to be a diabetic. In fact, we know that there's an increased risk of complications like delayed wound healing, and that depends upon your degree of hyperglycemia and also the duration of your hyperglycemia. So the worse your hyperglycemia and the longer you have this very poor hyperglycemia, the greater your chance of developing one or more of these complications. And why does this happen? Well, the current understanding is that the high blood sugar causes damage to biomolecules in your body. And this reaction is called glycation. Don't confuse it with enzymatic glycosylation when you're making glycoproteins. That's a highly regulated process. This is a spontaneous reaction. It's just sugars attaching proteins. And we've known about this reaction for a long, long time. It used to be called the Maillard reaction. It was discovered by a Frenchman called Louis Maillard. It's also called the browning reaction because it happens in food and it causes browning. I'm sure all of you have done some cooking and you've noticed it goes brown. Well, that's what's happening here. You're seeing the Maillard reaction. You're seeing the browning reaction. It's the sugars reacting with the protein and causing the browning. And you can see this physically because the reaction is happening so fast. You're using very high temperatures. But believe me, the same reaction is happening in your bodies, whether you're a diabetic or whether you're a non-diabetic. The only problem is it's happening very, very slowly. So you're all being cooked from the inside, okay? Okay, I know there's a large number of chemists in the audience, and I don't want to disappoint chemists. I've got nothing against chemists. I'm not a chemist myself, I'm a clinical biochemist. So this is what's actually happening at the sort of molecular level. Proteins have 3 amino groups in their structure, N-terminal amino groups, secondary amino groups, and lysine and arginine residues. And these can react with carbonyl groups from sugars in the acyclic form. So it's simply a reaction between carbonyls and amino groups. It's a nucleophilic addition reactions, and you form what is known as a shift space, which is freely reversible, and that happens within a couple of hours. These shift spaces can rearrange to a more stable product called an amidori product. And as you can see, the carbonyl group has been regenerated again. And that's what glycation is. The only problem is it doesn't stop there. This amidori product can undergo further oxidative reactions, very complex reactions involving lots of steps, involving intermediates like these substances here, 3-deoxyglucosones, and eventually what you form is what are known as advanced glycation end products, or AGEs. So these AGEs form on proteins inside your bodies, inside your body tissues. And these AGEs can be protein-protein cross-links, or they can be groups attached to proteins. They frequently cause fluorescence, they cause cross-linking, they often cause browning. In people with diabetes, because their average sugar levels are high, you get more glycation of body proteins and you get more AGEs. Now, I know I've shown proteins and sugars reacting here, but you can get these AGEs, you can get glycation of anything with amino groups on it. So it can happen to purine pyrimidine bases in nucleic acids, it can happen to certain phospholipids, so anything with amino group. It's all about carbonyl groups and amino groups. Again, just to please the chemists, some of these advanced glycation end products have been characterized. So this is one called pentozidine. It's a protein-protein cross-link. This is another one called purine, which is not a cross-link, but it's just got a fluorescent group attached to a protein. There's about 30+ different types of advanced glycation end products. The other thing is that what often happens is glucose can be converted to a molecule called methyl glyoxal. And this is a very reactive dicarbonyl compound. It's hundreds of times more reactive than glucose. And it can be formed from glucose, and it can also be formed as a side product from various metabolic pathways. And its concentrations in your blood, if you're a diabetic, are pretty high. It's about 300 nanomoles per liter. And normal people, it's about 100. So there's quite a high increase. And there's a lot of interest in methyl glyoxal because the current thinking is that a lot of the AGEs in our body are derived from methyl glyoxal. So where do these AGEs come from inside our bodies? Well, they can form endogenously on growth factors. They can form on nucleic acids, serum proteins, structural proteins, or they can come from outside our body. So if you happen to be a cigarette smoker, you'll probably have high levels of AGEs in your blood. If you eat processed food, McDonald's, Kentucky's, okay, Burger King, you'll have a lot of AGEs in your body, okay? So they can come in from the environment as well. And this is how they cause damage in our bodies. They cause protein-protein cross-linking, they can decrease ligand binding, they can modify half-life of proteins, they can alter the way the proteins are recognized by the immune system. Proteins have a precise structure that's important for their function. And here, that's being changed because of glycation, because of AGEs, and often what you find is that they lose their function. But also, during glycation, you can also make what are known as free radicals. Okay, so glucose, for example, this is a typical glycation reaction. The glucose can undergo auto-oxidation in the presence of oxygen and transition metals to produce free radicals. These Amadori products that I talked about can also generate free radicals and undergo auto-oxidation. And AGEs, when they interact with the receptors, which I'll talk about in a minute, can also generate oxidative stress. And free radicals, basically, I'm sure most of you know, are molecules with an unpaired electron. They're often referred to as reactive oxygen species. Most of them are derived from oxygen. And the free radicals themselves can cause damage. They can cause fragmentation of proteins, they can cause lipid peroxidation, oxidation of nucleic acid, and ultimately they can kill cells. So together with AGE formation, you're generating oxidative stress. And indeed, diabetes is a disorder associated with increased oxidative stress. More recently, there's been interest in interaction of AGEs with their receptors. We now know that there are receptors in our body found on endothelial cells, smooth muscle cells, and so on. And when AGEs interact with the receptor, that generates oxidative stress, and it activates all sorts of molecules that are associated with inflammation. So interaction of AGEs with the RAGE promotes inflammation, and that seems to underlie many of the diabetic complications that we find. And of course, there's a lot of interest in drugs that can inhibit glycation and reduce AGE formation. And in fact, there's 9 classes or 9 steps in the glycation reaction where a potential drug could act. So you could have drugs competing with the sugar or with the protein to prevent glycation taking place. You can have drugs that can break down Amadori products. You can have antioxidants, which can prevent formation of AGEs. You can have AGE cross-link breakers. You can have drugs that prevent interaction of AGEs with the RAGE, then RAGE signal transductors. So there's there's a hell of a lot of interest in this area to try and develop drugs that can reduce glycation, reduce AGE formation. Many labs are working on pharmacological compounds. Our lab, we've been particularly interested in natural substances. So a few years ago, we became interested in Momordica carantia, or Karela, as you can see here. And people eat this, they eat it either whole, or sometimes they just use the outer flesh, which is the green bit. Sometimes they eat it with the inner pulp, which contains a seed. Sometimes they eat both. And the very brave ones will actually prepare a juice, okay, a Momordica carantia juice, okay, and then they drink that, which is what I do every day, of course. Okay, so that's Momordica carantia juice. And there's been a lot of interest in Momordica carantia, or Karela, for many, many years. Historically, it's been used to control diabetes, okay, in many parts of Asia and many other parts of the world as well. And of course, nowadays you can get Momordica carantia tablets, and you can also get Momordica carantia juice available commercially as well. And it's got a lot of health benefits. Okay, most recently, it's been suggested that it can stop COVID-19 in its tracks as well. Some of the key ingredients. So there's been a couple of papers published very, very recently. But anyway, we've been interested in Momordica carantia in relation to diabetes. And there's a few things that we know about it. It actually improves insulin secretion. Okay, so that's one thing we know. It contains substances which lower your blood sugar. So polypeptide P is one substance, and the other one is carantin, and both of those affect your blood sugar levels. And quite a few studies have been done in patients, diabetic patients, and most of these show that Momordica carantia intervention lowers blood glucose levels. There are some differences in the various studies because some people have used the whole plant, some people have just used the Momordica carantia extract, and then there were differences in the duration of the intervention and so on. But by and large, most studies are showing that it's effective at lowering your blood glucose levels. And the other interesting thing is, Microvascular crantia promotes wound healing. This has been demonstrated in rats, mice, and in rabbits. If you have a wound, even if they've got diabetes, you can apply Microvascular crantia topically, either as a cream, as an ointment, or as a gel, and it promotes the wound healing. But we don't quite understand why and what the mechanism is. And that's one thing that we were quite interested in our laboratory. So we asked the question in our laboratory, first of all, this is going back a few years, what effect does Microvascular crantia actually have on the formation of advanced glycation end products? So we did some simple in vitro studies in our lab, and what we did was, let's just go back. What we did was we glycated a protein in vitro. In this case, it was lysozyme. We took it as a model protein. We incubated it with methyl glyoxal to make AGEs. And here is a gel, and this is lysozyme on its own. As you can see, it's a single band. Where we've got lysozyme incubated with methyl glyoxal, look at all the cross-linking that's happening, dimers, trimers, tetramers, and so on. These are the cross-linked AGEs that are being formed. But then apply increasing concentrations of Microvascular crantia flesh extract, okay, and see the reduction in the cross-linking. We can scan these gels, and we can see a dose-dependent effect. So as you increase the Microvascular crantia flesh extract, you reduce the formation of cross-linked AGEs. I'm showing the results for MCF here, but we've got similar results with pulp, and we've got similar results with carantin. So effectively, a Microvascular crantia extract is able to inhibit formation of AGEs. We then wondered what effect Microvascular crantia actually has on angiogenesis of endothelial cells. That's the sort of key process which appears to be affected in diabetic wound healing. So what we did was we looked at the effects of Microvascular crantia extracts, first of all, on proliferation of endothelial cells. That's one of the key steps in angiogenesis. And again, we use the Microvascular crantia pulp, Microvascular crantia flesh, and then the key ingredient, carantin. Okay, so we looked at the extracts of these. And we used a positive control, which is FGF2. And as you can see here, these are the initial cells, control cells, and then the FGF2 promotes proliferation. But if you look at Microvascular crantia extracts, then again, all three extracts are having a positive effect. They're all promoting proliferation of endothelial cells. We then looked at the effect of Microvascular crantia extracts on migration of these endothelial cells. So this is untreated cells. So what we did was we had a monolayer of endothelial cells, and we created a wound, and then we looked at the migration of the cells from the wound area, and we looked at the number of cells that migrated under this. So this is looking at the effects of FGF2, which is a positive control. So you can see lots of migration happening here. But then look at the effects of MCP, MCF, and then carantin, and they're all promoting migration compared to the control. So again, having a positive effect on angiogenesis. And the final thing we looked at was tube formation in angiogenesis. And again, we've got similar results. So this is what you're seeing with FGF2. It promotes tube formation, but then all three extracts at different concentrations, again, promoting tube formation. So we looked at tube formation in Matrigel, 3D Matrigel cultures. So again, these extracts were promoting tube formation. We then wondered what actually happens, and does Microvascular crantia affect intracellular signaling in endothelial cells? So we decided to do some experiments to investigate this further. And what we looked at was PIRK1 and PIRK2. So that's phosphorylated extracellular signal regulated kinase. So that's one of the signaling molecules associated with angiogenesis. And so we looked at the effects of MCP extract with signaling in endothelial cells. And again, what we found was that there was an increase in PIRK1-2 levels with the MCP extract. So we did this by Western blotting. We found same results with the MCF extract. Again, it sort of stimulated expression of peer-1, peer-2 in endothelial cells relative to the control. And we found a similar effect with the carantin as well. So it seems that Momordica carantia and its extracts are enhancing peer-1 and 2 signaling. We then wondered whether Momordica carantia are protected against AGE-induced inhibition of angiogenesis. We'd done some previous work, and what we showed is that advanced glycation end products inhibit angiogenesis. And this has been shown by our group, but also by some other groups. And maybe that explains why people with diabetes, why they have delayed wound healing. If you've got AGE circulating around in your blood, they may be inhibiting angiogenesis, and that's what might actually explain the delayed wound healing. So we've done some of this work, but we wondered whether the Momordica carantia extracts would offer some protection against AGE-induced inhibition of angiogenesis. So the first experiment we did was to look at effects of these extracts on AGE-induced inhibition of proliferation. So we've got our control cells, we've got our positive control, FGF2 here, but then look at the effects of AGEs. Okay, there's a significant reduction with regards to proliferation. But the 3 extracts appear to have a protective effect in the presence of AGEs. Okay, so all 3 of them increased proliferation. We looked at the effects of Momordica carantia extract on AGE-induced inhibition of migration this time. So again, AGEs, reduce migration compared to the control. And then again, the 3 extracts we found had a positive effect. And similarly, we reported protective effects of these extracts. Again, AGE-induced inhibition of 2 formation. So again, AGEs inhibiting 2 formation, but the Momordica carantia extracts are protecting against this inhibition. So this is was quite interesting to us because what it means is that if somebody with diabetes was perhaps taking these extracts, maybe that's how they're actually having an effect, and that's why you're seeing this protection against delayed wound healing in a lot of the animal model work that has been done so far. And finally, we wondered whether Momordica carantia actually acted via the receptor for advanced glycation end products, or RAGE, in endothelial cells. So what we did here was we've got an anti-RAGE antibody, and effectively what that does, it binds to the RAGE receptor on endothelial cells and blocks them. Okay, and we also use a isotype IgG1 control. So this is an antibody which doesn't bind or doesn't block the receptor, but otherwise has all the sort of characteristics of the antibody. And we did these experiments, and what we found was that whenever we blocked the RAGE receptor, we found that the extracts as well as the AGEs had no effect. So whenever we blocked it, so here it is here, and there's reduced expression of peer-1 and peer-2. And what that suggests is that the Momordica carantia extracts are actually acting via the RAGE receptor. Okay, so just to summarize the findings of our study, what we showed was that Momordica carantia extracts, they inhibit AGE formation, and we published this as a paper in PLOS One. We did all the mechanistic studies. We also found that the Momordica carantia extracts promote angiogenesis of endothelial cells, and that would promote wound healing. We found that they protect against AGE-induced inhibition of angiogenesis in endothelial cells. So AGEs have, they inhibit angiogenesis, whereas Momordica carantia extracts have a protective effect. And we also were able to show in these experiments that Momordica carantia extracts, they act via the RAGE receptor to alter intracellular signaling. And of course, these extracts might offer therapeutic potential. So for many years, people have been saying that, if you've got diabetes and you go on Momordica carantia, you get better control of your blood sugar. But also, there's anecdotal evidence that you get protection against diabetic complications. So there may well be some truth in that saying. So in conclusion, I think foods like Momordica crantia, they deserve more attention. They may protect against diabetic wound healing. These studies were in vitro. A lot of the other studies were in animal. Then we need to do more human studies to look at Momordica crantia to see if it protects against diabetic wound healing. So there's an urgent need to do more human studies. And of course, maybe we need to reconsider the advice we provide to diabetic patients. Treatment of diabetes is not easy. Drugs for diabetes, metformin and sulfonylureas, they're very expensive, even more so in developing countries. So you need to have something which is more cost effective. And maybe, using nutraceuticals, using things like Momordica crantia as part of your diet may have beneficial effects. And as I said, you need healthy food for a healthy life. And I really like this saying, let food be thy medicine and medicine be thy food. And I think that applies particularly to Momordica crantia. So maybe we should include more foods like Momordica crantia, even though they say it looks ugly in our diet. And of course, a healthy lifestyle is important for a healthy body. So if you look like this guy here, okay, and you're out of shape, then you need to change your lifestyle. You need to go for a healthy lifestyle, eat healthy food, plenty of exercise, and engage in healthy activities. And you might end up looking like this guy here. On the other hand, you might not want to look like either of them. Okay. So I want to finish off by thanking my group. Most of this work was done by Dr. Ali Al-Johi, who was my former PhD student. He's standing there at the back. He got his PhD and he's gone back to Saudi Arabia. He did most of this work. But also Dr. Sabine Matu Nasui, who's a postdoc in our lab, and Dr. Dong Hu, who is standing here. And also I had help from my friend, Professor Mark Slevin, who's an international authority on angiogenesis, so he guided the studies as well. And finally, thank you for listening to me.
Thank you, Professor Dr. Star, for a very nice presentation and also very important information for us. Now we have time for some questions. Yes.
In diabetes, there is dysregulation of macrophages. Due to it, there is delayed bone healing. So, if you want to have a proper bone healing, there should be activation of macrophages, especially M1, M2 macrophages. So, have you tested this extract on activation of macrophages and neutrophils?
No, we haven't done that. As I said, wound healing is a very complicated process and there are lots of steps involved. We've just focused on one step, which is the angiogenesis. But I believe there are other groups looking at the effects on macrophages, for example. Okay?
Thank you for the nice I want to ask you about as you know, the age is very important for the neurodegenerative disorders, especially the dementia. Because I worked in the RAGE for years. We have a medicine for phase 3. Unfortunately, it's failed at phase 3.
Okay.
So, but is there any correlation with the or is there any study that is being conducted the more the for the treatment of Alzheimer's disease on the age targeting or the RAGE?
Not that I'm aware of. Okay, there's a lot of interest in AGEs in Alzheimer's disease, and quite a few studies being done there. But I'm not seeing any papers on the protective effects of Momordica crantia. I've not come across them myself.
There's a direct correlation between the neuropathic pain, the diabetes, and diabetic patients, as you indicated, the COVID-19 also. Survival rate is dropping down with hyperglycaemia. So maybe it's worth to investigate. Is there a direct correlation with the treatment of So especially in dementia
Yeah,
and the Alzheimer's.
I'm sure it is. I know there are some, quite big groups working in Germany and looking at the effects of HEs in Alzheimer's disease. And they've been working on it for years. And I think they did the initial pioneering work, to demonstrate the presence of HEs in the Alzheimer's brain. But I don't know whether they're interested in natural products. Okay, there's only a few of us who are interested in natural products. Initially, most people were sort of focusing on pharmacological compounds. And I'm sure you know aminoguanidine, which underwent clinical trials, phase 3 clinical trials, but it failed. And the reason for that was because of the unacceptable toxicity. And so our group, we've tried to sort of focus on natural products. And a lot of other people now are, coming in this direction as well.
Very well, thank you.
How much Microvascular extract is recommended per day for the diabetic patient?
That's a difficult one. Okay, it really is difficult. I mean, we used fixed amounts for our in vitro studies, but I really can't answer that question. Some people just eat Microvascular fresh, like a salad, right? Some people maybe make the juice, a glass of juice, and maybe drink that a couple of times a day. You can always cook it, right? I mean, what my mom does is she takes the whole fruit, she takes out all the seeds, and then she fills it up with minced meat and spices, and then she fries it in oil, okay? And that tastes lovely. But I don't think it's good for you because I think a lot of the good ingredients inside there, they get destroyed when you cook it. Okay?
So you are the senior research, very comprehensive report and research you just to put up about with reference to diabetes.
Yeah.
So as we know, in our countries about 15% of diabetes, 2%. So my two questions is concerned. One is about when your research is to be tried any group of patient and control with reference to HB1AC.
Yeah.
Actually, one question. And second, as far as we know, the diabetes is the malfunctioning of the pancreas and do not utilize insulin and beta receptor cell. Have you any, just to advanced countries, have you any chance or is going on the transplantation of pancreas is going on, and what are the possibility of pancreas transplantation in Pakistan?
Okay.
Thank you very much.
Okay. So I'll address the first part of your question. Okay. Just remind me what it was. Sorry. Oh yeah, I know now. Okay. HbA1c. HbA1c. Okay.
Just a group of
Yeah.
people or
Yeah.
voluntarily or just
Okay.
examine and observe
Right.
with reference to HB1AC.
Okay, I get you. Right, this is what I'm suggesting. If someone becomes diabetic, what usually happens is they're put on medication, and the first medication tends to be metformin, sulfonylureas. Okay, that's what usually happens, and that tends to be very effective. And then what happens is after a period of years, the effects of those drugs decline. So even though they're on metformin or sulfonylureas, their HbA1c starts to go up, okay, and it continues to go up. And if this continues, eventually, about 50% of them go on to insulin injections. And these are type 2 diabetic patients. And that is really bad news, and you have to inject yourself twice. My suggestion would be that this small group of people who are on treatment, but who've still got high levels of HbA1c, this is perhaps the group that we should target for a nutritional supplement, something like Microvascular, to try and give them better control, right? So I think that group is important. Also, there are people who just don't have access to metformin because it's expensive. They're very poor, right? Again, maybe that's a group that we should be targeting as well with regards to Microvascular. Okay. With regards to your second question, I don't know about the recent work on pancreas transplantation. They've talked about it for many years, and there's been lukewarm success. But I'll tell you one thing: there's a hell of a lot of interest recently on reversing diabetes. It's known as a diabetes remission. That's a term that's been introduced a couple of months ago by Diabetes UK. And what's been shown in these studies is that if someone's got type 2 diabetes, you put them on a very strict diet, 600 calories for about a week, and that normalizes their blood sugar levels. Keep them on that diet for about a month, and effectively, right, their insulin production, sensitivity to insulin, etc., returns to normal, and they lose the diabetes. Okay, so they essentially become non-diabetic. But it only works for type 2 diabetics, and it only works for people who've been diagnosed with diabetes fairly recently, like the last 3 years or so.
Thank you very much, Dr. Nizar, for a very nice presentation. And here is with you. Now, our next speaker and the third speaker is Professor Dr. Rahman from Department of Biochemistry, University of and also from Dr. Rahman Center for Molecular Medicine and Drug Research, International Center for Cancer Pakistan. Dr. Rahman has research interest on insulin secretion mechanism and anti-glycation mechanism of microvascular beta subfunction and management of diabetes by natural. He has received NSC for fellowship from Ministry of Science and Technology, Government of Bangladesh, next fellowship of PhD studies on Government of Japan, and several grant and several research grants from different international organizations. He is acting faculty, Bangladesh University of Sciences, Bangladesh, and also educated in the European Institution of Study Diabetes, Japanese University in Bangladesh. The topic of his presentation is discovery of lead molecules from natural products for diabetic research. Now I invite Dr. Rahman for his presentation.
Thank you, Madam Farjana, for your nice introduction. So thank you very much for the organizer for inviting me. So I'm in a transition stage. I'm here and there and here. So I'm sharing some of my experience, what I did here. And when I came in Karachi, so this is my 29th visit in Pakistan. So a long time I stay here, so I feel it is more home. So even I just count this time. So I went my hometown less than Karachi. This. So thank you, Dr. Nasser, for your good introduction of the diabetes. So now it is a pandemic situation. If you, every day you can understand how many diagnosed, how many dead. But diabetes more than that. Every 2 seconds, one diabetes patient diagnosed. Every 7 seconds, one diabetic patient died. So Pakistan in the age of diabetes. So this is the recent data of Pakistan, Bakai, Bite, Karachi. So the number of diabetes in Pakistan, prevalence, it's more than India. Though it is number game, India more population, more number. If you consider the prevalence of diabetes, so this is the recent data from Bite, 26%, the prevalence rate is 26%. If you compare India per million people, the per million people diabetes is more in Pakistan compared to India. And there's a debate. So in Western country and our in Asian country, so in diabetes mostly either it is insulin secretion deficiency or insulin resistance. Those are obese people, they mostly suffer from insulin resistance. They are non-obese people, they are insulin secretion deficiency. So in Kyoto, this is the Kyoto declaration, that means in diabetes in Asian subjects, it is insulin secretion deficiency is more prominent than insulin resistance. We also got the same result in Dhaka. So all over the world, as Dr. Nisar Ahmed introduced the Mamadika Charantia, all over the world, people are using natural products. In our country, in this country, we take it as a first step. But in the Western country, they took it when no drug works, they go back to the nature. In Pakistan, 70%, all over the world, USA, UK, everywhere, natural product there. So this is the most beautiful place for me. I work here for many days, still I'm here. So what I did, as I told you, in non-obese diabetes. So first time in Pakistan, we developed a non-obese diabetes model with the pups, 2-day-old pups. We give injection, and after 3 months, what happens? It destroys initially the beta cell, then few beta cell is remaining. This few beta cell would make regenerate, that make insulin beta cell. If you see it here, this is the control, and this is the diabetic model. Insulin is there, but less of insulin. So the beta cell problem. So how this beta cell, the insulin or the insulin is produced from the beta cell. Because beta cell is the only factory for the production of insulin. So a lot of work has been done. This is the Mamadika Charantia, which simply we call it Corla. This is the most extensively studied, most extensively used for the diabetic patients, for the management of diabetes. So here we found that here, this is the contour, this is the diabetic, the islet size is increase of type 2 diabetes, size of islet is decreased. But when you give the treatment, the islet size increases. Then the islet size increase is due to the alpha cell or beta cell. We did immunohistochemistry. We did insulin, glucagon in the same. We found that this red one, the insulin, green one is the glucagon, and this is the red green insulin. Insulin is the outside, inside, glucagon outside. So when you give that, and we found finally that this Mamadika Charantika, it increased the beta cell. How it could be? It increased the number of beta cell, as well as it improved the quality of beta cell. In this way, it is the molecular mechanism. In this way, it improves diabetes. So another natural product, we call it amla. What happens here? If you see it here, this is the control, this is the diabetic number of islet size, as well as this one. These two doses, it increases the beta cell, as well as it increases the beta cell size. So once these natural products, so what is next step? How to do it? We got an invitation from Andhra. So one of my student, Mr. Abdul Hamid, at that time Mr. Abdul Hamid. So in Andhra, we have an organization in Dhaka. Sorry, Iqbal is the board member in this Andhra. So what I did, there is a technique we call islet isolation technique in the islets of Langerhans. So this student, I sent him in Andhra fellowship. I sent him Dhaka, and he learned this technique. And when he came back in Karachi, once we take different compounds. ICCBS is the hub. Many people are coming, isolation, purification, everyday routine of our colleagues as well as other colleagues. So this is the mice islets. This is the DIC image of mice islets, and this is the pancreas, and this is the mice islets. First we check it as islet. Okay, then we did immunocytochemistry. This is the islets immunocytochemistry, and this is the DIC image, and this is the triple channel immunocytochemistry. And with this, what is the insulin secretion mechanism is two types. One is potassium channel dependent, and another potassium channel independent. If you see, potassium channel, both are channels dependent, they are required for the insulin secretion. Once glucose enters the body, the ATP is increasing. If ATP increasing, potassium channel is closed. If potassium channel is closed, the calcium channel is open. This calcium channel, if it enter, it is the main factor that makes synthesis of insulin. This is the potassium channel dependent pathway. Another pathway is potassium channel independent pathway. This cyclic MP, this stimulate the PK as well as effective pathway, and this is also in this way it increases calcium at enhanced insulin secretion. So the compound we got in here, we did both the potassium channel and potassium channel independent pathway. What we did, this is the mice, this is the pancreas, we also did the pancreas, this is the partial purification one, and this we did with this islet, we crushed the islet, which is not in vitro, not in vivo, we call it ex vivo. And this islet, as we isolated freshly from the Langerhans, this is more physiological to do experiment rather than the cell line. So with this crest, this is the islets, pure islets. here islets, this contains the alpha cell, beta cell, everything. So with these islets, what we did insulin secretion activity as well as its mechanism. So till now, almost we did tested 1,000 compounds, and we got some no activity, some moderate activity, some potent activity. So almost 11 compound we got very, very good and very potent. With this potent compound, we focus on mechanism. So for considering both the potassium cell independent and potassium cell independent. The commercial drug available in the market, most available drug, we call it tolbutamide, glibenclamide. So this enhances insulin secretion, but the problem is it enhances insulin secretion at low glucose as well as high glucose. So if you have diabetes, high glucose. If you have high glucose, there is a problem, but you will not death. Death will be not occur. With 10, 20, doesn't matter. It will take a long time. But if hypoglycemia happen, death will be within seconds. This hyperglycemia is dangerous. Hypoglycemia is more dangerous. So this is the drawback of the drug. So we are focusing, can we find some kind of compound that enhance insulin secretion only at high glucose? With this focus, we got many compounds, but those compounds, this is the one compound, ED12, we found that at 3 millimolar glucose with the ED concentration with up to 400, it has no role on low glucose. However, high glucose, it enhances insulin secretion dose dependently. If you see right side, tolbutamide is BD1, and the low glucose, it enhances insulin secretion. But however, ED, low glucose, no glucose. Up to our glucose level is 5.5. Okay, at this stage, it has no effect. So this is interesting molecule. What we did then, we found in different pathway, and we found that it has no effect on potassium cell, no effect on calcium cell. It works on PK pathway. So we published the data in pharmacology. And then another compound, very simple molecule but interesting molecule, we call it coxazole. This is also same molecule. This is also similar molecule. It also enhances insulin secretion exclusively. Thank you. And you see plasma data. This is control cell. This is the coxazole. And if you use potassium channel opener and potassium channel closure, almost its current is inhibited. But if you coxazole and control, no effect, no potassium channel involvement. So similar here, we use verapamil. This is a compound that inhibit the calcium channel. If you control cell, and this is the verapamil, you see it here, almost inhibition. But if you add coxazole, control more or less similar current. That means this is not involved in the calcium channel. So then, what kind of possible mechanism we did? It's phosphodiester inhibitor that inhibit the cyclic AMP, and FSK, the cyclic AMP enhancer, and Antileishmanials cyclase inhibitor, PK inhibitor, MAC kinase inhibitor, and PKC inhibitor. We found here very interesting here. If you see here, this is the control data. When you add Antileishmanials cyclase inhibitor, the partial inhibition is there. PK inhibitor, partial inhibition is there. MK inhibition, partial inhibition there. PKC inhibition, this inhibition. But out of this four, this PKA and MAC kinase, this is the most predominant role. And we think that maybe this work in PK and ERK pathway. So then one of my collaborators in Japan, so this immunosynthesis lab, they have some. So we did insulin secretion activity at the after we call it after 1 hour. So what happens every second or every minute? We don't have this facility. So one of my collaborators, Izumi, so one of my student Abdul Hamid, I sent him to in there. Okay, he watched the poster. What he found here in coxazole and control, initial stage it has no effect. The low glucose, coxazole and control similar. What we did here, we found a similar better result there. Different low glucose, it has no effect. So only the high glucose, it is coxazole. This is the red one is the control and green one is the coxazole. You see, at the second phase and initial phase, low glucose, no effect. At this high glucose, it affects only high glucose at second phase of the insulin secretion. So from here, we conclude that yes, coxazole, it has no effect on potassium channel, no effect on calcium channel. It can affect on cyclic AMP, it can affect effect2 and both pathways. Recently, we published the data in European Journal of Pharmacology. So now, one of my students, Sabira Javed, so she's working on this in vivo kinetics of coxazole. So what we did in coxazole, we made some nanoparticle, with this coxole and nanoparticle, we did some in vivo pharmacokinetics. So we made liposomal based nanoparticle, silver based nanoparticle, and gamma tissue based nanoparticle. And with this, what we did, so and we characterized this nanoparticle. This is the coxole, normal coxole, and when we make nanoparticle, it's shifted. This is the characteristic feature of nanoparticle. So this is a very interesting result and editing data. What we did in this is the 50 milligram per kg. We put it coxole in the fasting rat, and after collection, we collect blood from tail vein at different time point. After collecting blood, we make plasma, and the plasma, what we did, we sent methyl acetate and methyl, we make it centrifuge, and with series filter, we did SPLC data. So if you see this is SPLC data, SPLC chromogram of coxole. So with different time, this is the standard curve of coxole, and we measure different time points: 0 minute, 15 minutes, 30 minutes, 60 minutes, 120 minutes, up to 240 minutes, different time points in the coxole. What is the, so if you see here, what is the concentration of coxole in the blood in the rat? You see it here in three different. This is the silver nanoparticle. This lower one is the only coxole, but when a silver nanoparticle, that means concentration of drug in blood is increasing. That means it is more availability, more absorption. Similar here, and over here in the coxole liposomal particle, you see it, this is the only simple coxole. But when you make coxole nanoparticle, this is the concentration of coxole. That means more absorption happens in the blood. This maybe coxole is good, as well as coxole nanoparticle is more far better. So next, we tested any of the two compounds. One of my student, okay, Mr. Khan, he's working on it. This very recent data. So we also found this compound. It enhances dose dependently. This is the ACM compound. You see this ACM. This is the control ACM at low 3 millimolar glucose. It has no effect. However, at TB, tolbutamide, this is commercial at 3 millimolar, it enhances insulin secretion. When we add TB plus ACM, no difference between two. That means our control, our group, this is our compound, has no effect of low glucose. But at high glucose, this is control, this ACM, the ACM enhances insulin secretion better than the tolbutamide. When we add it here, we have some additive effect. That means our compound, this ACM, exclusively enhance insulin secretion at high glucose. We also tested, this is the low glucose and cell line. We have mean 6 cell line at low glucose. You see the red one is the insulin. Here, insulin, this is the 200 millimolar glucose. So if high glucose, insulin is secreted, there is less strain. So here the control ACM and this ACM and control, you see it partial inhibition, ATP and total complete inhibition. So here the same result. So this is another study that verapamil, this is a part of a calcium channel inhibitor. When you add this calcium channel inhibitor, the insulin secretion, no insulin secretion anymore. No insulin secretion anymore. So recently, very recently, we got this data. So we did calcium fluorescence imaging of this compound. So one of my student Abdul Hamid, collaboration of Japanese, we did this experiment there. So we did calcium imaging of this compound. So what he did, isolated islets, monolayer preparation, and some fluorescent compound, and we did calcium imaging. And very interesting, we found that this is the ACM, this one the blue one ACM, and this is the control upper one. That means ACM has no either control and ACM. It is similar. They give the similar pattern of calcium imaging. That means it has no effect on calcium channel release. So if not, then what is the mechanism? Then we made the potassium channel independent pathway. In the potassium channel independent pathway, this is cyclic MP, PKA, PKC. This is the major pathway involved. So here I found it ACM. This is the high glucose. This ACM here. If we add IBMX, this is the inhibitor of phosphodiester inhibitors. It inhibits insulin secretion. This FSK, this is cyclic MP activator. So if we add this one with compound and without compound, FSK it makes this to this. But in case of FSK with ACM, it makes this to this. It has some role in insulin secretion. And this is another inhibitor of cyclic, another inhibitor of adenylate cyclase. This make it inhibition here. Then what we did, this is a PKA inhibitor. When we add PK inhibitor, we got very significant inhibition. Calcium, this is the PKS inhibitor. When we add, we got some partial inhibition. And this is another inhibitor we use. We also get partial inhibition. So very interestingly, the another found that those no one can inhibit completely. Then we use another inhibitor, though in the cyclic AMP it works both in PKA as well as effective pathway. So what we did here, we measure the PK inhibitor as well as effective inhibitor. You see, from here to here, it significantly inhibition the insulin secretion. So when you add both of them, this is significant but not complete. That means this pathway, PK pathway, effective pathway, these two pathway is involved in insulin secretion, as well as other pathways also involved in insulin secretion of this compound. So we also did some docking study with the help of Dr. Johid group. We found, yes, this compound has a strong interaction with PKA and some partial interaction with the PKC. From here, we conclude, yes, HCM stimulates insulin secretion only in high glucose, and HCM, the mechanism is possible mechanism with cyclic MP-PK and cyclic MP-PK2 pathway. This is the major pathway. Other pathway may be partially involved. So this is a very interesting slide for me. You see, there is airplane, 4 wheel, 2 wheel in the land, 2 wheel here. So there is a reaction in chemistry. There is a reaction we call it transition reaction, transition phase. So this is the airplane, 2 in the sky, 2 in the land. This I correlate it. Exactly happened to me. Okay, so I left Karachi on a leave on 28th February. I reached in Dhaka on 29th February, and from March 1, Karachi University shut down due to COVID. Okay, so then I want to come. Sometimes shut down here, sometimes shut down there, sometimes lockdown here, sometimes lockdown there, sometimes airport is off. So I'm waiting. So when because I feel more homely here. Okay, all the people, all the especially I love my students. I feel them. I like them. So my wife has a complaint. That means I sometimes forget my marriage day, my birthday, my day, especially. Okay, but I never ever. If he asked my student where he is now, within a second I can tell he's here, he's there, he's doing this. I can tell everything. But the big complaint for my wife. Sometimes I forget many things. So I learned one thing, Dr. Liaquat Ali. So whatever you have with you, please use it. Yudh mein baat hai na, jaisa des, aisa bes. So I worked for the, okay. So when decision is uncertainty is there, one private university, we call it Prime University. I joined there. So their private university, the research thing is very neglected. So what I did, so I have some idea, some here and some part-time experience. So tea, people, especially the Bangladesh, Pakistan, the people like tea very much. If they are very sad, have a cup of tea. If they are very good mood, cup of tea. Evening, morning, tea has no time. So I found one thing, the tea may be a good solution. So tea, I found in Japan, normally they are most healthiest, long, because after meal, they don't take water. They take ocha, different tea. So if you're taking water, it's nothing. This water, if you some natural products, maybe some flavonoids, anyone, they seasoning, they take it. It's something is better than nothing. In this after meal, so tea is enhanced insulin secretion activity. It's very true. Some evidence is there. But with the black tea, if you add sugar, the insulin secretion activity decrease. If you add milk, the insulin secretion activity decrease. If you add milk and sugar both, synergistic effect. So try to take black tea or green tea with milk, without sugar. Right? Follow Dr. Iqbal sir. So with this, so one of my friend, biochemist friend, he was working on tea and he did PhD in tea. So I discussed with him. So with our experience, a lot of spices is there, a lot of many things is there. So my experience, part-time experience with okay, what I did, normally the things we're taking every day. So some spices, some bay leaves, amla, tejpata. So some kind of leaves, spices as well as bread. So what kind of things? In many things we choose 11. Out of 11, with different combination, we make different combination. So my university is prime university. So my student what he did, different leaves, spices, or normally we use it normal life. Different, finally the moringa, mango, lemon leaf, tulsi leaf, and bay leaf. So this leaf in different combination permutation, we did make it this combination, we make it tea. And with this tea, we did, if we add A plus B plus C plus D plus E in this different combination, finally we made a tea, and we are focusing on taste as well as color. But different combination, we found a very good tea. And then with this tea, we tested in 500 normal or university staff. Everybody tasted. What is the taste? So it is good, but a lot of improvement is needed, and some experiment for antioxidant and as well as anti-diabetic activity is needed. We are focusing on it, and chemical analysis remaining. So this kind of work I'm working there. So prime. So hopefully this kind of tea can be made at iccbs. So instead of purchasing tapal tea or this tea, we can make a iccbs tea. This research as well as branding of iccbs. So when people came from outside country, we can get a souvenir. This is the best tea in Pakistan. Best herbal tea or best green tea. What is the difference? It is unique in a sense. It is scientifically evaluated, scientifically examined. Everything is written here. So these are the main students. So Abdul Hamid, Dr. Abdul Hamid, Dr. Ali Raja, he did lot of work, lot of report. Initially, up to midnight we worked day and night. Mashallah, we established this isolation technique and control it worked very well. And he did also in his student life. He visited many places, Munich, France, many places with this all data. And recently he come back from Japan to Karachi. And Ali Raja and Isra Khan, Isra Khan. Now he's taking care. He's taking care of this all the kinds of experiment. He's very active, dynamic, and ladies and gentlemen. So also I'm taking. So everything has a bad side. Everything has a good side. Corona stopped me to coming here, but I'm connected through different group, faculty group, PCMD group. What happens every day, at least I can understand. So Dr. Iqbal sir gave me some kind of opportunity to take class online with this my 702 courses. So I teach them and I give some feedback. So sometimes I found that yes, I can see everything with the CCTV computer. So whatever you are doing here, I can see from my home. Okay, who is coming, who is going, everything. Okay. So these students were from last year batch. They write a big, sometimes I told, okay, if I come here. But I told one thing, distance doesn't matter. I connected. So I found in the whiteboard in the PCMD lecture room, they write think and read. And last one, distance doesn't matter. Either you're anywhere. I think this is good what my student like my words. Okay, the students, are you here? Thank you very much. I am Dr. Hafiz. This is the first time you met, na? I am Dr. Hafiz. So this is my advertisement. My student Abdul Hamid, Dr. Ali Raja Shah, Isra Khan, Abira Khan, he did the lipid study and Faisal Khan personal study. My colleagues here, my student in Dhaka, and especially the ACC Pakistan, we got the fund and for the training. And we normally is a tea that my friend is working, and especially the Gunma University, Japan. So they did a lot of study, the in vitro kinetics, as well as they get support. They took one of my students, postdoc. Okay, we did calcium imaging and all the things is going on. Inshallah, we'll continue it. And so everybody has a model. I have some model. I have not only one model. I have 4 models. So Professor Mushid Jamal, so the chairman of NRAP, initial pharma chairman, and Dr. You know very well, Professor Chaudhary. Okay, I have nothing to say. So when I came here, when I met him in 1997 in Dhaka on a conference, at that time he was the associate professor. So everybody was surprised to see at the time he was too young. Still he is young. Okay, still he is young. So I was surprised. So then when I got an opportunity to work here, so I took this opportunity. Okay, and I think it is my best decision in my life. I came here, I work with him, I am working with him. Okay, and another thing is my mentor, Dr. Liakotali. So today I'm here because he ignited me. He ignited me. And Okinawa, Japan, this is the healthiest place in the world. So, but due to cold, it is the healthiest place for them. But cold, due to cold, I cannot enjoy this place. But this is the Panjani Center. This is so beautiful, no cold. To me, this is the most healthiest place for me. Thank you. Thank you. Thank you very much.
Thank you very much for your presentation and for sharing your experience. Now we don't have time for question and answers. So there is an announcement from our panel committee. After lunch, there will be parallel sessions at NJ in hall A, B, and C. Oh yes. Sorry.
Yes, please.
So after that, there will be parallel sessions at LEJ in hall A, B, and C, as well as in the auditorium. So our volunteers will guide you to LEJ hall after lunch, and the session will start sharp at 2 p.m. Now, all of you are requested to proceed to Golden Jubilee Hall for lunch. Thank you very much.
Ladies and gentlemen, may I have your attention? So before you proceed for the lunch, I do have a good news again. So there is a musical evening sharp at 7 p.m. at the same place, this auditorium. And this will be followed by the gala dinner at 8:15 in the lawns of Professor Ataraman Laboratory, that is right in the front of the auditorium. So you all are requested to join, and you, the foreigner participants, are requested to kindly wear your traditional dresses if you have. So it will be a cultural evening as well. और पाकिस्तानी मेहमानों से दरख्वास्त है कि गाला डिनर रजिस्टर्ड पार्टिसिपेंट्स के लिए है. अगर आपने किसी मेहमान को अपने साथ लाना है तो उम्मीद है कि आपने उसकी रजिस्ट्रेशन पहले से करवा दी. Thank you so much.
Thank you.